Now, let's get a closer look.
The trachea is the large airway that extends from the larynx and divides into two primary bronchi. Because the trachea and bronchi share similar functions, their walls are composed of similar tissue types as well, but with a few key differences. Let's first look at a low-power image of the trachea, stained with hematoxylin and eosin, or H&E for short. At this magnification, we can easily see the C-shaped cartilage ring, which is unique to the trachea. Also, you can use the cartilage ring to determine the orientation of the image, since the closed side of the ring points anteriorly and the open side of the ring will always face posteriorly towards the esophagus. The free ends of the rings are connected to one another by smooth muscle, called the trachealis muscle. The smooth muscle can be differentiated from the surrounding tissue by the long appearance of their cell bodies when cut longitudinally. Their lack of striations also indicates that the muscle is smooth muscle, not skeletal muscle. In older individuals, portions of the tracheal cartilage can transform into bone. For example, this cross-section of a hen's trachea shows areas of calcification, with some hyaline cartilage still present on the right. Looking at the epithelium at 10x magnification, it's easier to see the different layers of the trachea. The lumen of the airway is seen at the bottom left of this image. So the very thin layer lining the trachea's respiratory epithelium. Zooming in much further to approximately 100x, the cells that make up the epithelium can be distinguished more easily. Similar to the nasal and bronchial epithelium, the tracheal epithelium contains three main types of cells: ciliated pseudostratified columnar cells, the pale staining mucus-secreting goblet cells, and basal cells, which are found closer to the basement membrane. The basement membrane can be seen as a pink or eosinophilic band that separates the epithelium from the underlying connective tissue. Be careful not to confuse the layer of mucus lining the epithelium with the cilia on the surface of the columnar cells. The cilia is much smaller and only extends about 5 to 7 micrometers from the surface. The cilia in this image can barely be seen as the dark pink lines just under the thicker layer of mucus, which can be about 20 to 30 micrometers thick. The proportions of these cells will vary based on their location within the trachea. So the upper trachea will consist of more goblet and basal cells, while the lower trachea will have more ciliated columnar cells. The layer just below the respiratory epithelium is the lamina propria, which consists of loose connective tissue and is also highly vascular, as you can see by the large number of blood vessels within this layer. Moving deeper, the next layer is the submucosa, which contains many seromucinous glands that are less abundant in the lower trachea compared to the upper trachea. Oftentimes, you can differentiate the serous cells and mucus cells by how well they stain with H&E. Similar to this image of the submucosa, the serous cells stain much darker than the mucus cells. If it's not easy to differentiate the cells based on their staining, they can also be identified by their nuclei. The serous cells have round nuclei that are typically closer to the center of the cells, while the mucus cells have flat nuclei that are found against their basement membrane on the outer portion of the mucus glands. Between the submucosa and the cartilage ring, there's usually a very thin layer of fibroelastic tissue. The white space on both sides of the layer is just an artifact from preparing the slide. It's also normal to find this layer missing, and the submucosa will simply merge with the perichondrium of the cartilage ring. Alright, let's move on to the primary bronchi. From this low-power image, we can see that the bronchial wall has a few structures that are very similar to the trachea, although the supporting cartilage is now made of flat interconnected plates that surround the lumen on all sides with occasional gaps, as you can see on the right side of this image. The classic C-shaped cartilage rings of the trachea are no longer present in the bronchi. Looking at the bronchial wall under higher magnification, the epithelium is the layer on the far right, and the supporting cartilage on the left. If you remember, the lower trachea has fewer goblet cells than the upper trachea, and that pattern continues in the bronchi, where there are even fewer goblet cells present in the epithelium. Also, the ciliated columnar cells are not as tall. The bronchi also have a layer of smooth muscle between the lamina propria and the submucosa, which is responsible for bronchoconstriction. The bronchi will have noticeably fewer seromucinous glands than the trachea, and similarly, there'll be fewer glands along the distal portion of the bronchi. Alright, as a quick recap, the tracheal and bronchial walls share a lot of characteristics with one another. They both contain ciliated pseudostratified columnar epithelium with goblet cells, seromucinous glands, smooth muscle, and cartilaginous support. Their differences include the trachea's unique C-shaped cartilage rings with smooth muscle that connects the two ends of the ring, whereas the bronchi have interconnected cartilage plates instead. Also, the bronchi has shorter epithelial cells, fewer goblet cells, and fewer seromucinous glands.